Welcome again to our July session of Extension Hot Topics. Today's topic, Animal Disease Diagnostic Laboratory overview. We are very excited to have Dr. Burcham with us today to give us that background, especially for, I know a lot of the county fairs are going on with a lot of animals roaming around, so this is a very relevant case. I meant to say earlier as we were chatting before the Zoom started, I really want to show this to all of my vet sciences clubs because this is a resource that is very helpful and I hope to learn quite a bit. So, everyone, if you don't mind muting yourself, questions that you have throughout the talk, if you could put those in the chat box, we will do that. We are recording. It is now 12:02. The floor is yours, Dr. Burcham. I'm going to go ahead and mute myself, and any questions, like I said, that you guys have, just throw those in the chat box.
All right, thanks, Jeff. Appreciate the introduction and appreciate the opportunity to present today about what the Animal Disease Diagnostic Lab is. As Jeff said, I'm happy to be interrupted, so I don't know how well I'm going to keep eyes on the chat. Feel free to unmute yourself and ask a question too if you want to do that as well. So my name is Grant Burcham. I'm a veterinary diagnostician at the Animal Disease Diagnostic Laboratory, and we're going to talk a little bit about that. The background picture for the entry slide is where I work. I work at the Southern Indiana branch of the lab, which is located in Du Bois County. So I'm coming to you now from southwestern Indiana, and that's a picture out our lab's window of the hills, rolling hills of southern Indiana. So the laboratory, as I alluded to, has two locations, physical locations. So there's the main lab, which is located on the campus, the main campus of Purdue University in West Lafayette, Indiana. So recently renamed after a former dean of the veterinary college, Willie Reed. So it's the Willie Reed Animal Disease Diagnostic Lab. Got the address and phone number there and our web address. That's a picture of the main lab up on campus. We have a second location. This is in the southern part of the state, located kind of on the county line of Du Bois and Orange County. So if you're familiar with where Jasper, Indiana or French Lick, Indiana are, we're about midway between the two. This lab is located on the campus of the Southern Indiana Purdue Agricultural Center. So there are several, and I'm not sure if this has been a topic or not, but there are multiple agricultural centers around the state, 9, I think, and we're located on one of them. And that's our address, phone number, and web address. So that's the building I'm coming to you from currently, named after a former legislator, Dennis Hickey, from this neck of the woods. So what do we do? This mission statement and vision statement were taken from our website. So we want to provide diagnostic services essentially to a wide range of clients, animal producers, veterinarians, folks that need surveillance data, so regulatory agencies. We want to be the go-to lab in Indiana for diagnostic testing for veterinary species. That includes production animals as well as companion animals. And we have some role with wildlife species as well, working with those agencies, state agencies like the DNR that have purview over wildlife. So what is it exactly that we do? And I think these were specific goals that the ADDL has that I think are helpful with explaining to general population what it is we actually do. So providing diagnostic testing services that are prompt and accurate, using what we consider state-of-the-art equipment and facilities. We also want to disseminate that knowledge and provide expertise on animal diagnostics to veterinarians, veterinary professional students, veterinary technicians and nurses, the public, as well as everybody that needs to know about how to recognize and diagnose animal disease. So that's, we play a pretty important role in training future veterinarians about how to recognize and test for certain animal diseases. And then finally, we really want to develop and validate diagnostic tests that are needed here and throughout the country at large to improve on existing test methods, develop new test methods for diseases that we have, new diseases that come our way, which happens often, as well as those diseases that maybe don't have tests. A lot of toxic or nutritional diseases maybe lack tests, and one of the things we like to do is develop tests for those. So these are roughly our goals: diagnostic testing, which is a pretty important service, teaching future veterinary professionals, and then discovery or research into diagnostic tests. So I've got some examples of some of the services that we offer. So I'm not going to talk about autopsy or animal autopsy. We're going to go through a case actually later in the presentation to illustrate what that's like. But we also do parasitology, so fecal examination. Do my animals have intestinal parasites? How many and what kinds? We can tell you that. We do some bacteriology or microbiology. So that might be a question like, what caused this abscess on my animal? What antibiotics can I use to treat it? And broadly, not just abscesses, but any type of bacterial disease, of course. We do serology, which some people may know as blood work. We can answer questions like, how many members of my herd have been exposed to bovine leukemia virus? That's a question that we can answer with serology. Molecular diagnostics, this is the cutting edge, if you will, of diagnostics in general. So these are tests that examine for DNA or RNA of infectious agents. So it's not limited, for example, to just bacteria, but viruses, parasites, any sort of infectious agent that has DNA or RNA. So for example, we can ask the question, are these calves actively infected with rotavirus, for instance, or insert any disease name that you want. Also toxicology, we have a toxicology section too. So did my dog ingest rat poison? Is my sheep selenium deficient? Those are the sorts of questions we can answer with toxicological methods. And broadly, these sections are really based kind of around the method used to detect that particular pathogen. So here's a breakdown of the different services at the two different labs. Here at the lab I am at, we don't do molecular diagnostics or toxicology, but pretty much everything else. And if we can't do it, we'll send it to the main lab in West Lafayette. So the ADDL, to put us in kind of a national context, is part of a network of animal health laboratories across the country. Many of those labs are located in or at institutions that have schools of ag or colleges of veterinary medicine. And so University of Illinois, Michigan State University, for instance, two of our peer institutions that have veterinary diagnostic labs. But we have a national network of those labs. There are federal labs as well. So our federal reference labs located in Ames, Iowa, that would be the National Veterinary Services Lab. And then we have a lab formerly located at Plum Island off the coast of Long Island on the eastern seaboard. It's recently been moved to Manhattan, Kansas, and that's the Foreign Animal Disease Diagnostic Laboratory. Those are the labs that test for diseases that we don't have here in the United States that we really want to know if we get, because these are diseases that can have really high health consequences for animals and economic consequences as well for those commodities that the animals produce. So as being part of this National Animal Health Laboratory Network, or NALN, we, the ADDL, do some testing for some of these high consequence foreign animal diseases. We've got them listed here that the animal health officials really need to know whether we have these or whether we're free of these. So one that's recently been in the news a lot and that we have spent a lot of time on is the highly pathogenic avian influenza, or bird flu. This is a disease that was considered a foreign animal disease, still is, but we have currently now within North America and have been dealing with for the past few years. All right, I'm going to talk a little bit about what to do if you have a diagnostic question for livestock or companion animals, for instance. First thing, contact your veterinarian. If you have an animal pass away or die, keep the animal cool if you can. In the refrigerator is best. Freezing is the second best option if you can't get it to us for a few days. You can come to the ADDL. You don't need an appointment. You can certainly call ahead and ask questions if needed, and then fill out a submission form, and we'll take it from there. And I like to think of the ADDL working as a team with a farmer and the veterinarian, or an animal owner, not necessarily a farmer, but an animal owner and a veterinarian, all working in concert to help the animal or the herd understand, diagnose animal disease. So we're all from Indiana, right? We all like basketball, presumably. So just think of it as a team approach to helping the animal and animal health. So this is what we refer to in the veterinary field as the veterinary client-patient relationship, right? So a veterinarian should have a relationship with a client and the patient, the animal itself, right? And so the ADDL really wants to support that relationship and make sure that that relationship continues. This has become even more important in recent years because due to federal law now, it's really necessary for veterinarians to have the VCPR in order to prescribe certain types of antibiotics, for instance. So a veterinary health team approach is really what we want to support. We want to support your veterinarians. We want to support you and help animal health overall. So we're going to switch gears a little bit, and I'm going to present a case, a diagnostic case that I worked up. This has been a few years now. It occurred in the fall of the year. So a little bit of warning ahead. I am going to show some pictures that show tissues from cattle, so some organs from an autopsy. So do know that that is coming here. First, I'm hopefully going to make this video work. So this is a cell phone video that was captured by a client of the ADDL. This is a beef heifer that's part of a rotational grazing operation in southern Indiana. This was the fall of the year, getting to be the fall of the year. And what you'll notice in this particular animal is she has her head down and neck extended. It's a little grainy, but she is open mouth breathing. You can see that her mouth's open. And there's quite a bit of foam around her bottom lip there. So she's breathing very heavily. This animal died overnight, and the producer brought her to us to do an autopsy. So that's oftentimes where our diagnostic cases start, not always. Sometimes we get samples that are unconnected from the animal, from a biopsy that your veterinarian took, or blood that your veterinarian took, or a swab that your veterinarian took. But oftentimes, especially when I'm involved, my training and background is as a veterinary pathologist. So when I'm involved, oftentimes there is a whole animal that comes to the lab for autopsy. So given the clinical signs, pretty worried about respiratory disease in this particular case. And so this is the opened chest cavity of that heifer. So the head's to the right, tail's to the left, and these pink structures are lung lobes. And you might notice that there's areas where it's really pink, and there's areas where it's kind of white. And if you get a little bit closer, here are those pink areas again, and you can see that those white areas are actually air. And so there should be air in the lung, right? But it should be within the air sacs. And whenever you see air that's outside of those air sacs, which would be the pink area, that's emphysema. So this is emphysema within this heifer's lungs. So that's air that's outside of where it normally should be in the lung. And that can occur because of really forced respiratory effort, where air will rupture air sacs and come into interstitial tissues. So this is a cut section of the lung. The rest of the lung becomes very collapsed and firm or rubbery. And then there's air within the interstitial spaces. So this is another section of emphysema, illustrating emphysema in this particular heifer. So not good. This is why she died. She had pretty severe pathology throughout the lungs. And it's difficult to tell here because you can't see the whole thing, but on at least this cross section, most of the lung is pretty severely affected. So what's the next thing we're going to do? We're going to look at that under the microscope, right? So a lot of my day is spent collecting tissues on the autopsy floor and then looking at tissues under the microscope that we've prepared. And this is a way where we can learn a lot about the process that was happening and then hopefully get some idea about what caused that process or initiated that process. So we've got 4 sections of lung here. You might notice there's a lot of clear space in some of these, and that's what you should see. There should be a lot of air within the air sacs, but then, for example, this one and other parts of these other sections are pretty dense. There's a lot of pink there, and there shouldn't be. So if you get a little bit closer, you'll notice that each of these should be an air sac, each of these little kind of open structures. And air sacs should have very thin walls because the blood flows through capillaries within those walls, and there's gas exchange between the center, the lumen of the air sac and the blood. It's how we exchange oxygen, how our red blood cells can pick up oxygen from the air. And here, those air sac walls are really thick and lined by really plump-looking cells. There's also cells in the center of these air sacs. And so you can imagine that there wasn't very efficient air exchange or oxygen exchange occurring at this interface. This is a type of interstitial pneumonia that we refer to in pathology as diffuse alveolar damage. So the alveoli are the air sacs, and so we've had diffuse damage of those air sacs in this lung. So that's helpful for me as a pathologist and a veterinarian. I can then think about and read about those causes of diffuse alveolar damage in cattle. And I did recognize this lesion grossly. I didn't need the microscopic exam to recognize it, but saw it grossly and made a search throughout the rumen. That's the beginning part of the GI tract in ruminants. So in cattle, the rumen is kind of the large part of the stomach. So it's called the forestomach. That and the reticulum and the omasum are the forestomachs where fermentation takes place, fermentation of forage and other plant material before it then moves to the true stomach, the abomasum, and then through the rest of the intestinal tract. Anyway, looked through the rumen because there are some toxic causes of diffuse alveolar damage in heifers. And I found some leaves that look like this. So as some of you may know, cattle don't chew their food super well the first time it goes down, and then they regurgitate it and chew it further, chewing their cud, right? So sometimes it's very helpful if you're looking for toxic plants to look in the rumen because the cattle might not have chewed it all the way. So I found some leaves that look like this, jagged edges, mostly green, and stems that look like this, right? So these, you might recognize that these stems are square. They're not round like a lot of different stems. And if you know about plants, you might know that anything with a square stem is part of the mint family. And so there is a toxic plant called Perilla mint, Perilla frutescens, that has some toxic components for herbivores, right? And specifically, they can cause respiratory disease in ruminants. They cause that diffuse alveolar damage because that toxic component is metabolized further within the lung, actually, within cells in the lung. And so that can cause lung damage. This particular plant is got some characteristics listed there. It flowers kind of in the late summer and fall of the year, and that's when it's nice and green and it looks really good. So it's one of those plants that towards the end of the year, other things start to look dry and not so great, but Perilla really comes into its own in the end part of the growing season. And so in this particular case, the animals were on a rotational grazing system, and this happened, I think, a day or two after they'd been moved to a new paddock. There wasn't Parliament in the new paddock, but they looked back at the other one, and maybe they'd spent a little bit too long in that paddock because the grass had been grazed down, and there was a dirt pile and a disturbed area, and there was a big stand of Parliament growing there. And at least one animal, this one, really grazed that stand of Parliament down the day before they were moved to that other paddock. It's interesting. This particular plant is marketed as shiso, or known as shiso. It's an invasive plant in North America. It's native to Asia originally, and it's used in Asian cuisine and it is flavorful and very aromatic. If you crush it, like many of the mints, it has a really nice aroma and a good taste, lends good flavor too. So this is from I think Johnny's Seeds maybe, website, and they're selling shiso. That's probably how it came to this country, was through, escape through people that are growing it for culinary uses, culinary reasons. But in small amounts, apparently, it's not bad for people. I haven't found any human intoxications in the literature, but poisons are all about dose, right? And so animals that are grazing heavily or eating a lot of it can succumb. Here's some pictures in the spring, pretty small. In the fall, it's looking really nice. Sometimes has a purple sheen to the underside of the leaf and those jagged edges. So very much what we found in the rumen on that particular animal. So in this case, we didn't need to do any bacterial culture or molecular diagnostics or anything. We could match what we were seeing in the animal to what we found in the rumen. We could potentially have a toxicologist identify the plants, for instance, and come to a diagnosis that way. So just an illustration of how we work up a case and how things can come through the diagnostic lab and hopefully come to a resolution that can then be used to help other animals on the farm or other animals at home, help their health and bring peace of mind to a producer or an owner. So this is a nice segue, I think, into a project that we had recently here at my lab here in the southwestern part of the state, at the Hickey lab on the SciPac campus. We, working with Jason Tower, who's farm superintendent here, Keith Johnson, former faculty member in agronomy up on campus, as well as Molly Hassenauer, local extension educator, we developed a toxic plant exhibit here at the lab. So this is meant to be a little bit of a living library, if you will, for producers that come in or people that are concerned about what they might be seeing in their pasture at this time of year, for instance. So we get producers that come in and they say, well, I've got a lot of this particular weed in my pasture. I'm not quite sure what it is. Or we might talk about a toxic weed and they say, well, I don't really know what that looks like. We can go out to the toxic plant exhibit and take a look at it. So got a couple pictures here to show the layout. Each of the plants, I think we have 24, is in a hard plastic ring that's sunk in the ground and has a little sign with a placard on it that you can use to identify the plant. There's also a QR code on there that you can use to learn a little bit more about the plant. So I don't know if anybody has encountered this particular book or not. Christy Mandan, the author and illustrator, I believe, of Millie Fleur's Poison Garden. This is a great picture book for young people and older people too. I find it really enjoyable. My daughter really loves it. Gives it 2 thumbs way up. And there's a sequel too called Millie Fleur Saves the Night. But the premise is that Millie Fleur has a garden of plants that are a little bit unusual, and her neighbors don't appreciate it a whole lot until they get to know a little bit more about it and what makes the plants unique and interesting. And so I like to think of the ADDL's toxic plant garden a little like Millie Fleur's poison garden, right? And the Poison Garden series, well, 2 books now, hopefully there'll be more. The Poison Garden series is based a little bit on a real life poison garden in Alnwick in Northumberland in the UK. There's a really interesting subset of that garden. It's a larger garden. It's not all poisonous plants, but a subset that is poisonous plants. And these images were taken from their website. A super interesting place. They've got some very toxic plants, some that they have under lock and key and only in behind bars, for instance, so people can't touch them or anything. And I think you can only gain admission to the garden with a docent at certain times, so you're not just free to roam at will. Makes me a little nervous for our garden. There's nothing around it, so hopefully we don't have anything as poisonous as they do, right?
Can I just a quick comment? For those who are master gardeners, this when you look at how they planted these plants, if you ever want to do a garden with displays, you bury the pot. And it is one of the best ways to keep things in check. Because I have put in weed gardens and different things where you have the identification. So just wanted to throw a shameless plug. If you do want to do a display garden, this method is just about as good as you can get.
Yeah, and that's a great point. That's why we did that, try and keep them contained a little bit, right? Because some of these are really invasive weeds and all of them were taken from from SciPac or other locations around the state. They're growing wild, all of them, whether they should be or not. It's many of them are invasive species and shouldn't be, but we have them and they're part of the landscape now, so need to know about them. But yeah, that ring keeps them contained. And it actually doesn't have a bottom. It's sunk in, hopefully to keep stolons and rhizomes and other things from spreading. This is an example of one of the plants, buttercup, for instance, and you can see the name there and the QR code and then the flower. So you can, depending on what time of year it is, for example, now the poison hemlock and the star of Bethlehem and the crest leaf groundsel, those guys are all done. And you see the bare stalks, and then other things are growing really well. So it's, again, a living library. So at your time of year what in our area, what's growing and what's happening. So if you do the QR code, you're going to come to a site that looks a little bit like this on your phone. And we've got general information about the plant, the concerns. So if, the time of most concern, compounds within that cause an issue, part of the plant that's most toxic. And then if you go down, there's another tab for livestock. Talk about which species are affected, whether it's pasture, stored feed, or both, clinical signs that you see. And then, if known, sometimes we don't really know this, but if known, the amount that's needed to be consumed for clinical signs or death to occur. So you'll know, hey, is this really poisonous? Is this only poisonous if they eat a lot of it, for instance? And much like Millie Fleur's garden, I think there's a lot of interest and beauty present in the SciPac poisonous plant garden, right? So these are all images from the garden. Thorns are part of Carolina horse nettle there, a solanum species. So this is also a flower from Carolina horse nettle, and then ripe berries from that same plant. Really photogenic plant. Top middle, Senecio species. So these will cause liver disease, chronic liver disease in animals that consume this over time. Top right hand is cocklebur. Again, a liver failure, more acute this time in animals that ingest the leaves of the cocklebur plant. Then everybody probably recognizes this down here because of the animal that's on it, right? This is a larva of the monarch butterfly. So it's a monarch caterpillar on the host plant for that species, which is milkweed. Milkweed contains a cardiac glycoside that will cause heart failure. All right. So we're wrapping up here. Just want to leave you with a few take-home messages about the ADDL and ways that you can learn more about us and get a hold of us if you need us. We're also happy to talk on the phone and answer questions, of course, related to animal health and diagnostic needs, right? How do I figure out what's going on? So this is a screenshot from our website, mobile compatible, has a lot of information about what to collect and what samples are needed for each test, as well as the turnaround time and the cost, shipping information to get that to us, or you can always stop by and bring the sample in. You can also use this. We've got a client portal for obtaining your test results, paying online, etc. So addl.purdue.edu. I'd encourage everybody to join our mailing list. You can scan this QR code, or you can go to the website, and there's a box here where you can click on and sign up for our newsletter and email alerts. So I'd encourage other people or everybody to do that if you're interested in the ADDL or have animals or interested in veterinary diagnostics. So you can click on which email list you want to join, but if you're like me, you're going to click on all of them, right? Get all the email alerts. So this is us, phone number for West Lafayette, phone number for the Hiki lab in southwestern part of the state. We've got some emails. You can email ADDL at Purdue or ADDL SciPac at Purdue, and someone will get back with you if you have a question. Again, my name is Grant Burcham. That's my email address, gburcham@purdue.edu. If you have questions or comments or just want to say hi. I really like where I work and find it really a photogenic place. So got some photos from the Southern Indiana Purdue Ag Center in different seasons. This might be my favorite one here. Fall's really beautiful in Indiana, I think. That's it for me, Jeff. So if we've got questions, I'm happy to take questions or engage in discussion about the lab and what we can do for people.
Yeah, no, that works out great. If you have questions, feel free to throw them in the chat box or unmute yourself. I did have one question in terms of year averages. What would you say the biggest type of samples you collect on average are, or maybe just from the last year or so? Because I was looking through your website, which I encourage all of you to go to the website. It's very thorough. But what's your biggest I guess specimen intake for your lab?
Yeah, we probably do between the two labs, we're doing between 1,800 and 2,000 autopsies a year. So that's a big specimen type. Uh, we also get a lot of blood, of course. So blood comes through the lab for serology testing. And then swabs are really important too. So especially given avian influenza, right? So a lot of times we're getting oropharyngeal swabs is the best way to do that in birds, in at least gallinaceous birds. Taking a swab of the inside of the mouth and submitting it to us will be a really accurate way to diagnose highly pathogenic avian influenza or bird flu. So I'd say swabs and blood and then whole animals are what we get. I also should say too, we do a lot of biopsy work at the lab. And so, for example, if you have a lump on your family dog, for instance, and take it to the veterinarian, and the veterinarian says, "Hey, I think that should come off." They're going to take that off, and they can send that to us, uh, preserved, and then we're going to make sections of it, look at it under the microscope, and give your veterinarian a diagnosis, like, "Hey, this is a benign tumor," or, "Hey, this is one to watch," or, "You got it all," or, "You didn't get it all, you need to go back." So we actually do a lot of biopsy work too, and so getting parts and pieces in the mail is also something that we do quite a bit of.
I will say thank you because I do have a cat. Her name is Carol, and she had a lump, and my local veterinary said we were going to send it into Purdue, and it was a good service to have. So, it does work. How many people do you have on staff with all those samples coming in, can you kind of give us the scope of your lab? And did you have technicians, or are you the guy with the gloves on? Are we taking away from your time with animals right now, or
Yeah,
what does kind
I
of
will I
the
will say after this, I'm going to don some gloves and coveralls, and I'll be in the back cutting things open and collecting samples. So we have at the main lab in West Lafayette, we have about 70 people that work there, and down here at the Southern Indiana lab, we have 7. So so probably between 75 and 80 people usually are working at the lab. That includes several veterinarians on staff. So I'm a veterinarian myself, and there's another veterinarian here that works with me. There's several up at West Lafayette. And so we're veterinarians that have done some additional schooling beyond veterinary school. Many of us have done some advanced training that might look like a residency program, right? Where you spend 3 years studying to take a board exam in a specialty. So my specialty is pathology. We have veterinarians who are specialized in microbiology and veterinarians that are specialized in toxicology, for instance. So we have multiple veterinarians that have gone on for additional training to specialize in those disciplines. So we have multiple veterinarians. We have several technicians that also work with us in our various sections. So we have technicians that go back on the autopsy floor and help back there. We have technicians that are doing bacterial culture and identifying bacterial species. Also technicians that are running blood work and also running those swabs through the molecular diagnostic instruments to come up with those results. So kind of a group of technicians, technical staff, and professional staff in the veterinarians. And then we've got a lot of support staff too that are up front talking to clients, bringing samples in, getting samples labeled appropriately and where they need to go. So cutting slides for us to look at those biopsies. So there's a lot of moving parts to it, and I think for what the lab is, we do a lot with the folks that we have.
Yeah, so I'll just some of my background. I've always worked with the plant and pest diagnostic lab, the animal
Yep.
side. Sorry, you guys were in the vet building. I was just one block over in the horticulture greenhouses. So
Hmm.
it's amazing what goes on. Would you say that, and I can if you guys have questions, just feel free to jump in. Would you say you do more livestock and commercial production, or would you say it's more pet? I don't know how you would distinguish that. Um, companionship pets, or is it how's the
Hmm.
breakup?
Yeah, the term you're searching for is companion animals. We usually refer to dogs, cats, and horses as companion animals, and then production animals, cattle, swine, sheep, goats, poultry. We do a lot of both. At the lab here in southwestern Indiana, we're doing mostly livestock and poultry, and that's why this lab grew up because of the heavy poultry populations that are here in the southwest part of the state. So we do a lot of poultry here, as well as other species, but we're probably like 85-90% livestock and poultry here. In West Lafayette, they because they're the proximity to the teaching hospital, they do a lot of companion animals and a lot of companion animal diagnostics up at that lab. So much more heavily tilted towards companion animals at the West Lafayette lab. I think at least for autopsy, the breakdown was about 50% large animals, and that includes horses, and 50% small animals, dogs and cats. So probably over 50% companion animal, at least coming through the postmortem floor up there. A lot of the swabs we're getting are from swine operations and poultry operations. So a lot of the molecular diagnostics are geared towards livestock. And then, of course, the biopsy service is, 95+% dog and cat.
Ah, awesome. Well, if there are no other questions, I did put the post-program survey in the chat box. I'm going to stop the recording. But before I do that, I am going to share my screen here, um, real quick. Again, we thank Dr. Burcham for being with us for the program. We have his information. August 21st, this is going to work really well to where we're going to continue to talk about toxic forages because they are an issue with all of that. And I'm going to put up, give me 1 second, folks. I'm going to Try to do two things at once. Getting difficult. Here we go. So if you guys can see the extension hot topics there, for the rest of the year, we are always taking different I'm losing my words, suggestions for topics. Courtney, I'll get those squared away. If you guys don't mind doing that post-program survey, Dr. Burcham, thank you again for your time, and everybody have a great rest of your day.
Thanks, Jeff.
Oh, I need to know. This is going to be on the recording now. Okay, now we're good.